and you have time to rest. Ready for business. We've been struggling and talking about spending much time on in terms of evolution is a wide course. And to God be the glory, we are coming towards the end of it. And we are just coming 4 weeks to the end of this program. And I'm trying to see if we can actually get to genetics and move a little bit deep into genetics, in as much as we might not, but the key words, we all know that most questions are coming in terms of test crossing. We'll get to that place, the first place. Now, genetic evolution, like you said, has so much on it in terms of the theories and the fathers of evolution, and how researches were made by all the scientists trying to prove what they come up concerning the development of organisms or the formation of organisms, either from non-living things, simple things, simple organisms to complex organisms. And also, after that complex, in terms of evolutionary classifications, and different classes, both plants and animals, whereby we also show that vertebrates, as an example, where they evolutionally believe that everything started from, that all of them appear from the same plane in terms of their development. And one word is showing similarities. When we looked at evidences of the position of a truth, when going deep to look at into those things, we might believe what they said, or we see the truth on some of the things they said. Then that leads us the last, before we get to microevolution, that is evolutionary trends or classification of evolution, starting from the simple ones. And we know that most of the simple ones have to do with the prokaryotics, then getting to eukaryotics. Those ones are developed from the single cell, or those that don't have, or multicellular cells. And when seen, it got to protistas. Now, protistas, you know, there are many microscopic organisms. And we have the plant part of them, we have the animal part of it. And the only thing they're made up of one single cell. And not that they're exactly looking like those plants and animals, but they have one or two features that have to do with animal, and one or two features that have to do with plants that made them to be classed under microscopic plants and as well classed under microscopic animals. Then in terms of trending, like I said, starting from the prokaryotics to the eukaryotics and then to the protistas. When we now get to the protistas, we know after the protistas we have other ones you cannot class them under animal, neither can you class them under plants. And that results in areas where we have the fungi. Am I saying something now? And the other ones under microscopic, we can still not class them under animal, you can still not class them under plants. And as well, they are called the monorans. Do we still remember that? That had to do with bacteria and co. But that one apart, they just stand out like that as apart from the fungi and other forms. Then when branch out, we know that the protists, we have the protozoans, we have the talophytes and so on, until you start getting to, from there, in terms of plants, the talophytes, getting to the bryophytes, from the bryophytes to the pteridophytes, from the pteridophytes, we have the skeletophytes, where they finally divided from the angiosperm and the gymnosperm. We still remember those ones. Then, coming to the other aspect, we now have the animals, where you have the protistas. From there, you get to the sponges. And in that aspect, you now find those ones that are a little bit simple, coming to the sponges, the what you have, the polypheras. After that, what you have, colytritas. From the colytritas, what we have, the platyhelminthes. From the platyhelminthes, what we have, the nematodas. From there, we got to what? Annelids. From the annelids, we now get to the molluscs. From there, we get to the arthropods. And now the products will have about four classes attached to them: the insectants, the crustaceans, the arachnids, and then we'll have the mirapods. Then we'll have the centipedes and the millipedes. And from there, that leads us to the echinodermatans. Now from the echinodermatans, if we still follow it deep, we now have what we call the chordatas. From that chordatas, we have the arthrosus. Then we now have the vertebrates and the invertebrates. We have mentioned the invertebrates, now coming to the vertebrates, which starts from the Pisces, or the fishes, and the amphibians, and the reptiles, or the reptilians, and then to apes, and then to the mammals. So if, as me, you're asked to, especially in practical, you might see them, when you are being given a lot of species of organisms, and you're asked to arrange the specimens A, B, C, D, E, depending on the number of species. It could be in the lines of animals, it could be in the lines of plants, and they are asked to arrange it in evolutionary trend in terms of you have to start from the simple. Did I say something now? You have to start from the simple, depending on what you are given. Depending on what you are given, it could be plants, and they'll ask you to arrange those specimens in terms of in an evolutionary trend, or classify them according to their complexity. And classify them according to their complexity. You have to start from the simple one. Did I say something now? Except if there is an instruction from starting from the most complex down. And that is why when there's any question, you read and pay attention on the questions following instructions given to you so that you don't miss it. Did I say something now? So from there leads on to the topic we have today, which is microevolution. The question is, what is microevolution? Does it mean that there are evolutionary trends, evolutionary organisms that are microscopic in nature? Yes, there are. But these ones are based mainly on diseases. Diseases that you cannot actually say how it came about. The abnormalities that occur within the body system of animals, especially when it comes to human. You cannot actually say, how did this come about? And you know that most of these things are carried mainly in the red blood cells. And we know that the red blood cells becoming red, actually in its normal nature, it is not red. But it contains a pigment that is red in color. And that is what gives the red blood cell its red color. And that pigment we call hemoglobin. Am I saying something now? Now when we say microevolution, that has to do with some diseases. Diseases like sickle cell anemia. To me, one cannot really say this is how it started, but it's like an infection that occurs in the hemoglobin. And we now represent this disease in terms of using the letter X. Am I saying something now? So we have some of these blood groups that are found in the surface of the red blood cell. So they are contained in the hemoglobin. So the abnormal ones contains this X, and which is the sickle cell, while the normal ones contains A, as it were, also in the hemoglobin. But each infection that result to this disease they call sickle cell, they are now termed with hemoglobin X. I might say something now. Being found there, you can only notice it microscopically. That result to this dangerous disease, and it is inheritable. It is inheritable. It doesn't really have a cure. The cure is when it comes to the mixing or combination of characters or genes, whereby you have a carrier that's having X or XX, the disease, and someone that does not have it at all, that carries AA. So when you have the two come together, the offspring that will be coming forth might have become a carrier, but as it were, it does not show much. And they even have a strong immune system than even the ones that have A. I hope you understand what I mean. So that is what microevolution is all about. So let's write down microevolution. I want to believe I've just said something a little bit to my own understanding. Answer me. Yes. Good. So as we write, to be able to explain the more microevolution. Example is sickle cell anemia. Please have a picture of it. Disease caused by the formation of abnormal hemoglobin known as X. Instead of normal hemoglobin A, instead of normal hemoglobin A. First of paragraph, hemoglobin is a protein of a complex molecule. Hemoglobin is a protein. Hemoglobin is a protein of a complex molecule made up of amino acid. Hemoglobin is a complex molecule made up of amino acid. First off, what hemoglobin A? What hemoglobin A? Please make sure you write it this way. What hemoglobin A and S? What hemoglobin A and X molecules are alike. What hemoglobin X, A and X molecule are alike except in one amino acid. Except in one ONE amino acid. First off, I repeat myself. Hemoglobin is a protein. It's a protein, a complex molecules made up of amino acid. Both hemoglobin A and X molecules are alike except in hemoglobin, or except in one amino acid. First off, the difference in structure. The difference in structure is due to a mutation in the gene. The difference in structure is due to mutation, or due to a mutation in the gene for hemoglobin. I repeat myself. I said the difference in structure is due to a mutation in the gene for hemoglobin. First off, Another paragraph. In the mutated gene, mutated, I should spell it? I should spell it? Who said there's M-U-T-A-T-E-D. M-U-T-A-T-E-D. In the mutated gene, comma, one of the base codes, C-O-D-S, one of the base codes in the DNA, one of the base codes in the DNA that should read R-E-A-D as the amino acid comma glutamic acid comma has changed its structure. Glutamic acid comma has changed in structure. Has changed in structure. Has changed in structure, comma. Hemoglobin X is less soluble. Hemoglobin X is less soluble than the normal hemoglobin A. Hemoglobin X is less soluble than the normal hemoglobin A. First of all, another paragraph. The hemoglobin X molecule tend to clump causing the red blood cells to become sickle shape. I said hemoglobin X molecules tend to clump, causing the red blood cells to become sickle shape. This results in severe anemia. This results in severe anemia. First off, now what are we not trying to say? Already we are meant to understand that typical example of a macromolecule or a macroevolution is sickle cell anemia, which is a very strong or fatal disease that tends to affect human. And these results were found in the red blood cell, which contains hemoglobin. And more especially when the hemoglobin becomes abnormal. Instead of carrying the normal A, blood apparatus will start finding X in it, which we cannot say how it results in death. But that's a kind of abnormality that we found in it, contained even in the DNA. Said that it resulted as a result of mutation. Who remembers what is mutation? Definition of mutation. Who remembers? Okay. Can somebody remind us? It is. I expected everybody to say it. It means like the chorus. Alright. So Say it loud so that they will hear you, please. One to go. Let me hear you. Okay. Genetic makeup. What is this? Taiwo, let me hear you say it. The sudden change in genetic makeup of an organism. Alata, let me hear you. Huh? There must be an idea. Open your nose and say something there. Oh, there must be an idea. Because we have come across this word several times. We came across it for the first time when we treated duration. I think I'm correct? Oh, yeah. Kadi Roger, say something about it. Huh? Stand up. Nobody should tell me no idea in the definition of mutation. It should be like a chorus. Even if you're sleeping and somebody tapped you, you say something about it. Oh, yeah. Sudden changes. Of an organism. HGS. Remind me your name. Huh? Oh, yeah, did you say something?
Mutation is abnormal changes.
Are you sure? Mutation.
It's sudden change.
Are you sure? John, no, I'm sorry. What? Okay. Of an organism. I think are you?
Mutation is the sudden change. Mutation is the sudden change in the DNA structure of an organism, which brings about new variants.
God bless you. Thank you.
Mutation is the sudden change in the genetic makeup.
Mutation is the sudden change in the DNA structure.
Are you sure? You are not listening. That's why I left all those information. Adama. What is man? We have been saying some things. Okay. Let's continue. This one is still coming. So let's continue. So we're trying to tell ourselves that the differences we notice in the structure is due to mutation in the gene for hemoglobin, whereby we saw some abnormalities in the hemoglobin of the red blood cell having a form of X, and why the normal thing should have been A. And we said it's a proteinous structures that are supposed to be found there. So whether be it A, whether be it X, molecules, they are alike, except in the areas of when we have one amino acid. Since we know that they contain protein, and amino acid happens to be the end product of protein, isn't it? So when we say it is a mutated gene, one of the basic codes in the DNA that should have been clear or should read as amino acid or glutamic acid will not have a different thing found in the structure. And exactly that is what actually results to that. And already we know that the red blood cell is soluble substance. But we found out that instead of being in a normal form in its solubility, because of the presence of the X, the solubility becomes so wide to compare what you actually need to see in A. And that will result in the weakness becoming too big. Am I saying something now? And when that is, it will affect the bones, where we have part of the bones like the bone marrows that are involved in the production of the red blood cell. So if the hemoglobin contains have this molecule that is so soluble, so the thickness to maintain, and that's why when you see some people having this problem when there is an attack everywhere, even the bones cannot sustain to carry or kill the person, like we said. Oh God, I know in the olden days when they started having such, they say now anyway. In the East they say no punching, but these are the things heritable. Or how it comes, or how it actually comes. I don't know. Everybody has already told us, I've mentioned, but these are the things they found in their research, found in the hemoglobin. In the hemoglobin. Remember that the antigens, antibodies, where did they find the antibodies? And then antibodies. Are you sure? Are you sure? Antibodies. Antibodies on the surface of what? Blood. These are things that by now on the not blood platelet. Where do you find the antigens? In the surface of the red blood cell. Antibodies are found where? On the surface of. God bless you. These are the things you should not forget. These are the things you see on objectives. Little thing. And some of you have collected from jam. And once you're trying to think and think, it should be on your fingertips by now. Because we mentioned it a lot, even when we're talking about blood transfusion. About some of the things needed for help that where variation played an important role in human. These are the things. Go back to who are those talking there? What was the last mention there? We said hemoglobin X molecules tied to clone, causing the red blood cell to become sickle shaped, and this results in severe anemia. Stop. Paragraph. Hemoglobin formation is controlled by a pair of genes. Hemoglobin formation. Hemoglobin formation. Hemoglobin formation is controlled by a pair of genes born on homologous chromosomes. Born on homologous chromosomes. You came across this word for the first time. Where? On which topic? Huh? Have you done genetics? Have you done genetics? Is that what? Cell division. Another which topic? Mitosis. There is a mitosis. More first, we came across it in mitosis. And we said that the genes appear in pairs, and each pair is called homologous chromosomes. You remember this diagram? You remember the diagram? Huh? And I believe you can level it. I believe you can level it. That's what. Objective. I believe you can label it. Huh? So a pair of a gene like this is known as homologous. One of it is called what? It's known as what? One of this is called what? God bless you. Sister chromatid. Chromatid. These are the things you should know. Yes, it's sister chromatid. If you don't want to, at least you remember it's chromatid. And when you have it split, at what stage? That will not split. At what stage of the mitotic division? At what stage? Yes, I'm referring us back a little bit so that will come out. At what stage? At what stage? Faith, at what stage? Who remembers at what stage? Huh? God bless you. Please help me to clap for him. It's like he's the only one that has been knowing all these things. So, at later faith, where it is formed where? I didn't wait to God bless you. At the center. And they are being held by what? God bless you, spindle fiber. Please, these are the things so that when you see them in question, it will not look as if you have not done it. And these are the simple things. Okay? So hemoglobin formation is controlled by a pair of genes born on the homologous chromosomes. Full stop. These genes are represented by the symbol. They are represented by the symbol HBA. They are represented by the symbol HBA. Represented by the symbols HBA. Full stop. A person with a sickle cell anemia has a pair of mutated hemoglobin in HBX. I repeat myself. Hemoglobin formation is controlled by a pair of genes born on homologous chromosomes. Full stop. These genes are represented by symbols HBA. Full stop. But a person with a single cell anemia has a pair of mutated hemoglobin in HBX. How many of us have seen a question like this? First question, you have. And there's a question that will still meet here, might not be in the person, that they ask in 1 year. HBA in carrier and the non-carrier. And that is why. Many people could not solve them because they didn't get to this point. So they don't understand what is HBA. When there is HBA, and HBA, it's a normal hemoglobin. But when you have HBS is a standard sickle cell anemia. But when you have HBX, that's a carrier. I hope you understand what I'm saying. In terms of, that's a carrier. But when you get mutated, when the gene makeup is just like that, you not having this. But the normal thing we supposedly will have without any infection is this. If we are not microevolution. These are the ones that are true. Whether we believe now in evolution or not. Did I say something now? Because these are things that are found within the body system of the human. They cannot actually say how it came about. Or research got it. And probably the same person that got it was able to put it like that, following this way. I don't know, don't answer questions. Are we okay? Paragraph. Sometimes a person has one normal hemoglobin gene. Sometimes a person has one normal hemoglobin gene and one mutated one. One normal hemoglobin gene, which I have mentioned, and one mutated one. So sometimes a person has one normal hemoglobin gene and one mutated one, which is HBS. Such a person is said to have the sickle cell traits. Such a person is said to have the sickle cell traits. Such a person is said to have the sickle cell traits. Full stop. Paragraph. People with sickle cell traits do not show any ill effects. Ill. Do not show any ill effects. People with sickle cell trait do not show any ill effect, although a small percent of their red blood cells shows sickling. I'm too fast. I should repeat myself. All right. I said people with sickle cell traits. That is, they are sickler. They are carriers. Not that they are full sickle cell. They don't show the traits. You will not even know. I hope you're not writing that one. You will not even know that they have the trait in them. You will not even know that they are AX. Let me use that one. You won't even know that they are HBA and they are carrier. The gene is HBS and HBA. You won't even know. There's no sign of effect. They hardly fall sick. They hardly fall sick. Their immune system most of the time is very strong. So it shows no ill effects. They show that they are even having the traits in them. So that's what it means. The people with sickle cell anemia do not show any ill effect, although a small percent of their red blood cell, shows sickling. A small percent of the red blood cell shows sickling. Full stop. Under paragraph, people with sickle cell anemia usually die before they reproduce. People with sickle cell anemia usually die before they reproduce. Full stop. What does that mean? Before they fully mature. Or maybe when they get married. They die very early. And that's why in times of counseling that Veneration showed us, we have to go for this test and make sure that the trait is not in. If you have this trait, please don't go for any person that have HBA, HBS. Go for HBA, HBA. That is it's in terms of genotype. Two of you has been talking. Two of you know. Kilogenesis. They're showing me your teeth. I know it's white. I said people with sickle cell anemia usually die before they reproduce. Full stop. Under normal condition, the harmful sickle cell genes should be eliminated. Under normal condition, the harmful sickle cell gene should be eliminated from the population. Should be eliminated from the population. Full stop. However, in parts of Africa where malaria is common, however, in parts of Africa, or most parts of Africa, where malaria is common, a high percent of the population has the sickle cell traits. However, in parts of Africa where malaria is common, a high percent of the population has the sickle trait. Has the sickle trait. Which means what they're trying to tell us that most areas, we know that majority of Africans who have this malaria character found inside of us. But God has given us the grace to actually stand it one way or the other. So once we take the antibiotics, it deals with them. But because of things around the mosquitoes, it kept on increasing. So because there's so much they believe in their research that most Africans, because you're immune or we suffer from malaria a lot, the majority of the Africans in our blood contains this sickle cell trait. That is, we have HB, HA, HBA, and HBS. That is, the trait is found inside of most African populations. Anyway, anybody knows paragraph. This is due to the fact that people with sickle cell traits are more resistant to malaria. Are more resistant to malaria. So due to the fact that people with sickle cell traits are more resistant to malaria than the others. But people with normal hemoglobin, you know what we mean by hemoglobin ABS, hemoglobin A. Normally, people with it are susceptible, SUSCEPT, IBLE, are susceptible to malaria. And those so affected usually die before becoming adults. Full stop. What they're trying to tell us is that even the people with the sickle cell anemia traits withstand the malaria attack more than the people with the normal. In as much as they suffer a lot, that's what we said most times. This is off record. Alright. What was the last sentence?
Adult.
Before becoming adult. Full stop. People with sickle cell anemia also die young of severe anemia. People with sickle cell anemia also die young of severe anemia. Full stop. Only with people with sickle cell traits usually live and reproduce. They usually live and reproduce. Full stop. We've already said that people with sickle cell anemia usually die young, so they might not live to reproduce, that is to give birth to offspring. But those with the traits, they live long and they reproduce. Full stop. As a result of this, the mutant hemoglobin gene that hemoglobin gene though harmful, is naturally selected in this region, that is in our region, is naturally selected in this region because of the immunity it provides against malaria. Because of the immunity it provides against malaria. First of all, another example of macroevolution is the selective breeding practice. Another example of microevolution is the selective breeding practice. Selective breeding practice. Who will give me an example of a selective breeding practice? Yeah? Selective breeding practice. We have mentioned that of let's say human, a sickle cell anemia. What of selective breeding? Trying to bring up something? Huh? Inbreeding. Crossbreeding. Okay, so inbreeding. What? What can you do in China? You read it all like okay, what happened?
They want a particular trait to continue their family. So they don't marry
outside. So that's an example of inbreeding. Yeah? Okay. Who has another example? Breeding practice. Crossbreeding, like you don't know what is crossbreeding.
When two different
Organisms.
yes, with different characteristics.
Okay.
And let's say one character of this one you want it,
What
and the
more than
second one you don't want it,
okay.
and another one has the one you
A better
want
one,
and you want
yes.
to cross together.
Alright. I know it's common among these dogs, people are training.
Yes.
Yes. Okay? That can be very easy as a practice. So that is it. So take note of it because you'll come across it. I'm sure if you're already good at fast questions, if you have started going to fast questions, you would have come across this. And that is under dihybridization. You mentioned hybridization. We have monohybridization, hybridization, and dihybridization. So we'll see come across them. Any questions so far? Any question? Any question? No question thus far. Yes? Genotype. Okay, let's go to genetics. To grow. It is possible. It is possible. By possibles. So much. That I cannot fully answer. But those in the medical line will give you answer. But I know that since they say it's so good, most times they don't grow. It takes gods. Medically, they keep infusing blood, removing blood. Although I heard that in America now, they can remove all the blood and return. They will remove all that SS, HBS. As they're removing, they're infusing new one. So that one will now continue. abroad. See, Nigeria can do it. I am telling you, the best medical doctors they have in America from Nigeria. Not one, there are many. The medical doctors they have abroad, they are saying it, they are Nigerians. The best computer analyst in the whole world, even in America, is a Nigerian. So we are capable of doing everything. We have the best everything. But because not equipment, they don't want to recognize anything if there's a word like that. So let's go to the topic genetics. Isn't it? You came across what? Scientific study. Okay. Scientific study of hereditary and variation. Does anybody have any other definition of genetics? Does anybody? So generally we might tell ourselves that genetics is a science of hereditary. Genetics is the science of hereditary. So in its definition. So it can be defined as the study of hereditary and variation. Genetics can be defined as the study of hereditary and variation. So let's look at the principles of genetics, what they account for. What are the principles of genetics? Accounts for. So the principles of genetics, what they account for. One, they account for the diversities of organisms. I would say it's a driving force. It's the driving force behind evolution. So we said that genetics happens to be a branch of sciences that studies hereditary and generation. And when we tend to look at the principles of genetics, we said is the diversities, or it gives account of the diversities of organisms, different organisms, the origination and how it came about, both living, both plants and animal, microscopic in their nature. So the principle of genetics accounts for the diversities of organisms, and also that genetics is a principal driving force behind evolution. It is the main driving force behind evolution. It is the study of genetics that disproved whatever the evolutionists came up with in terms of formation of organisms. So we said they are the driving force behind evolution. It also made us understand that genetics accounts for the adaptation of a species to its environment. How species are able, or different organisms are able to adapt comfortably and reproduce comfortably in their habitats. It is the cause of the principle of genetics. We also make to understand in the study of genetics, it helps us or explain that it is sexual reproduction that provides the opportunities of a species to adapt to its environment and as well evolve. So these are the main principles. We find that we have done adaptation, we have done evolution, and also in terms of the differences we find in organisms, it is genetics as well that brings up or gives us an eye opener on what it's all about. Now that leads us to the pioneers. Who are the pioneers of genetics? Who are the people that came about or proof or disprove what the evolution said? So that leads up to the pioneers of genetics. That is the fathers, those who came across that. Am I communicating? Am I communicating? Yes, I wasn't talking to myself. Was I talking to myself? So let's look at the pioneers of genetics, or the fathers of genetics. Pioneers of genetics. Now, when we're talking about evolution, during the Charles Darwin's evolutionary theories, he was able to know that the differences we notice among organisms as a result of the variations is as a result of what is being inherited from parents to offspring. But how that thing came about, he cannot prove it. So it was during the research of what Morgan brought up, Mendel, what's his name? Mendel. Look at what Charles Darwin was talking about in terms of characters. He now realized that the main factors responsible for those differences and variation is as a result of gene combination between combination among parents that shows forth in offspring. So he disproved most of the things that the evolutionists came about, and that results to the pioneers. In 1806, let's write down, continue. In 1806, an Austrian monk. I've asked us once here, who is a monk? Thank you. I think we have answered that, we discussed that. In 1806, an Austrian monk known as Gregor Mendel Who is asking me who is a monk? That's an assignment for you if you don't know it. No. You know you cannot serve 2 masters at a time. We have dealt with that. We even crack jokes on it in this class. I believe I'm not lying.
Yes.
We discuss it and also discuss none. N-U-N. You and I. So find out from your friends. Because that day I knew you went somewhere. Eh? Good. You can't serve 2 masters at a time. One suffers, the other will might gain. Might. So an Austrian monk, known as Gregor Mendel, laid down the basic law of genetics. Laid down, L-A-I-D, the basic law of genetics. Full stop, that is in 1806. Then a Danish botanist, known as what? It's like some of you know it. Called the factor that transmit disease, that transmits characters. Gregor Mendel was able to find out or carry out a research that most of the characters observed in offspring is being transmitted from their parents. And from his research, which he started or he did in 1806, in 1909, a Danish botanist known as Joachim, I think I mixed the spelling, called that factor that brings about the transmitted characters of as gene. Mendel proved that the characters were transmitted, but he didn't mention how. But this man, who is a Danish botanist, carried out his own research and found out that that which was responsible for the transmission of characters from parents to offspring is what he called it gene. That factor that is responsible for the transmission of characters from parents to offspring, he called it gene. In his research he did was in 1909. Remember, in 1806 was Mendel, who is an Austrian monk living in an Australian monastery where he stayed, carried out his own research following what Charles Darwin was saying, who could not dictate or say how the variations came about, found out that it was a transmitted characters from parents to offspring that shows the differences, notice the variation, notice between parents and offspring or offsprings of the same parents. But still in the research of other genetics found, this one, joiners, found out that that factor that is responsible for what Mendel said in transmission of characters, he called it a factor. And that factor that is responsible, he commonly named it as gene. and his research he carried out was in 1909. Then another person, a genetist known as Thomas Morgan, and that is in 1912, also found out that this gene of a truth that was called as a factor by journalists was carried by chromosomes, which is found inside what? The nucleus. Is that clear? So that these are the three major elements that are involved in genetics that are trying to give us what is all about. So I said, in 1806, an Austrian monk known as Mendel laid down the basic law of genetics. Then the Danish botanics journals called the factor that transmitted Mendel's character as gene in 1909. First stop. Then in 1912, an American genetist Thomas Morgan. An American geneticist known as Thomas Morgan, from his own research, that the genes were carried by the chromosomes. From his own research, found out that the genes were carried by the chromosomes. First one. So let's look at transmission and expression of characters in organisms. Transmission. How are these characters? Remember we said in 1806, Thomas Morgan, in his research, found out that the characters that are transmitted from parents to organisms is as a result of combination. And in 1909, another geneticist, which is a Danish botanist, known as journalists, called the factors that are responsible for transmitting characters that has been concluded and found out by Mendel, said that factor he called the gene. And not only that, Thomas Morgan carried out his own research and found out that genes are being carried by the chromosomes. And remember that we said that the chromosomes are a pair of contracted characters carried by or found in the nucleus that carry hereditary characters that tend to transmit from parents to offspring. So it was Thomas Morgan that carried out a research in 1912 that the genes are found in the chromosomes. And it is that gene that brings about the transmission of characters from parents to offspring. And there was a geneticist that carried and found out that. And what is the name of the person? It's not Thomas Morgan. That the genes transmit characters from parents to offspring. It's not recommended. It's not Charles Darwin. Joannes. He was the person who found out that what was responsible for transmitting the characters was gene. Mendel tried to disprove what Charles Darwin said. believed that characters you see in offspring was transmitted from parents to offspring as a result of combination. So he brought out the basic, the main law guiding transmission of characters from parents to offspring. Now, once he did that, the other scientists who are geneticists as well followed suit. I wanted to find out what is actually responsible from that characters that are found in either mother or father that is seen on the hybrid. Are you getting what I'm saying? Good. And that factor, he called it gene. That is actually the gene that is responsible for the transmission of that characters that Mendel found to offspring. Now, that is Janus. Then Thomas Morgan, in his own research, it's okay. There is a factor that I found inside that thing that transmitted the character. What is that factor? He started carrying out his own research. And we know that even from the organelles, there is an organelle called nucleus. And these things we are talking about are found inside that nucleus. And the major thread-like structure that is found there is called what? And the chromosomes is known in its character for carrying hereditary traits. Am I saying something now? Now that hereditary trait is what is being during combination of the two parents by sexual reproduction. By sexual reproduction. And we saw those factors that we see on the two parents in an offspring. Remember, he has called it gene. Now, in his own research, that's Thomas Morgan, he found out that those genes are found inside that thread-like structures. But the main person who started this hereditary variation is who? And that is why he was called the father of genetics. But others had to follow suit to actually confirm what it is. And they found other things inside. The factor called gene, and that factor called gene was carried by that thread-like structure, which is called what? Chromosomes. And the person that was responsible in finding it is what? Thomas Morgan. So that is why they are called the pioneers of genetics. They are just three of them. But the main first person, and that is why he was called the father of genetics, is what? Mendel. And he carried out a lot of research, which we want to look at now. How did Gregor Mendel find out that? This one just followed what Gregor Mendel did. He used a lot of things. He used plants in transmissions of characters. How did these characters appear in the offspring? That brings about what we want to talk about now, that have to do with reproduction. Remember we said that genetics in its study explained its sexual reproduction that provide the opportunities of a species to adapt to its environment and as well evolve. Evolving cannot come without combination most times. Did I say something now? Shoyein, are you very sure? Are you very, very sure?
Yes.
So we'll get to that combination. That is when we talk about transmission and expression of characters in organisms. So we want to see what Gregor did. So that tomorrow, if you want to say why is guy was so fair and the brother is not fair. How do we have different species of hibiscus flower? We have red hibiscus flower. We have white hibiscus flower. I have seen peach hibiscus flower. Pride of brothers, there are many. We have seen the red, seen the yellow, we have seen the white, we have seen the yellowish red, like you said. I've seen the peach, and I've seen the whites. These ones. But one funny thing when we want to talk about the expression that Gregor Mendel used in trying to explain this, he used a pea plant. And we'll get to know, by the time we meet tomorrow, we'll find out what was the reason, why should he use a pea plant. And there's another thing he used commonly, probably, although it was my high institution that mentioned, that's like Drosophila. Drosophila is like a fly. I don't know how many of you know Drosophila. Very small. You see, if you look at orange now, let me tell you, all of you know it. If you look at orange and drop it here, just give it roughly 20-30 minutes. If you come there, you see this something like reddish flies. They'll just fly. Reproduction have taken place there. It's so fast. It's so fast. So in his studies, sometimes he used that one. But mainly the major thing that was used was a green pea. Or let me say peas. Pea plants. We'll get to know the reason why Mendel used the pea plant to carry out its study of variations, how it is being transmitted, the characters from one organism to another. Any questions so far? We'll continue from there tomorrow. Please, I beg us, try even what we've done under that evolution. Look at questions, objectives, questions there.
Yes.